So this project is focused on how ocean currents affect diversity of plankton protists. So it's a project that has been involved, like we have been 2 physicists, Simone Pigolotti and I, and 2 biologists. That is Alex Busek and Tom Bourguignon, also from the university. So it's a interdisciplinary project. The origin of this project is this paper here that was published on Nature, Ecology and Evolution that basically analyzes the samples of the Tara Ocean project. That is a very huge project in which many groups, research groups, are taking samples from many, many points, as you see here, like these yellow points in the ocean. They take samples of water and they analyze everything there. Temperature, depth, salinity, everything. They publish everything online, and especially all the sequence data of all the microorganisms that are present in the sample. Everything is published there. So what they do in this paper particularly is to measure one of the main measures for characterize diversity of ecological communities. That is the species abundance distribution. So basically what you do is to measure the frequency of the abundance of a species. So basically the number of individuals that each species have. They count this abundance and observe this pattern. They observe basically 2 regimes. One is these black dots, and another is these red dots for high abundance species. They focus on this rare biosphere, as they say. Rare because it's very small number of individuals per species. And there are many, many, many of these individuals, very small abundance species. What they observe is that this is characterized by a power law with a very high exponent, an exponent that is higher than 1. Why I say 1? So the main theory to explain, the simplest theory that has been proposed in ecological area to explain ecological communities is the neutral theory that predicts that this decays as a power law of 1. This explains many, many, many communities. Although it's super simple, it describes many communities. However, it doesn't describe this system. And we want to know why. So there are not any satisfactory model that explains this. And what we want is to try to model it to understand what is happening there. Our hypothesis is that oceanic currents, due to these special structures like chaotic advection and the barriers that it creates, will affect the diversity. And so that the species abundance distribution is steeper. From doing that, we propose a model in which we have the plankton dynamics based on neutral theory, so that any individual is better than other. So it doesn't have, each species doesn't have a better fitness than other. Everyone has the same chance to die or reproduce, independently on the fitness. Because what we want to focus is on the role of the currents, not on the role of the dynamics. So we wanted to keep it simple. I see the effect of the currents here. For doing this, what we do is to do the dynamics for just a sample in the ocean. So we have a sample of a species that comes from a huge community, and we just simulate what happens with the sample that we are taking nowadays back in time. So we reproduce or trace back in time all the dynamics of such sample. So we reconstruct the ancestry tree, the family tree, until the most common ancestor. In this way, we have many, so we don't have to simulate the whole community and then just analyze one of the population, but just we analyze just the relevant information we want. So for the currents, there are many models that reproduce quite well the main characteristics of the chaotic advection of the currents, of the oceanic currents. We have taken this model as the main model. It's described in these two papers. And basically it's based on a jet here that we see. Particles goes here, but eventually they get trapped temporarily here, and then they goes back. So you have these idols here. But the particular model, we will show that it doesn't matter. The only thing that matters is that we have chaotic advection. We have proved that with other two models that I will describe later. But this is our main model. For the plankton dynamics, we base on a neutral model explaining this, in which we don't have the lattice as we usually have, that simplifies everything, but we have continuous space. That is what we need for our fluxes. So we call it forward model because it's forward in time. And basically it's, as many of you know, you reproduce, one random individual reproduces in some neighborhood with size L, then individuals diffuse, and then they die by competition with others. So in mean, on average, you will have always one particle per neighborhood. The problem, as I said, here is that you need to simulate the whole community. That is huge. So for avoiding that and do it a more computationally optimal simulation, and being able to do this simulation because you couldn't do it in other way, is to do a backward model. So from the sample backwards in time. So again, you have displacement or diffusion that is going to be backwards on time, the diffusion, and what we call coalescence. So basically you establish every time a particle enters the neighborhood, you establish that that particle is the child of the one that is here. Once you save that relation, you remove the particle, and then you start removing the particles until one is there. That one will be the common ancestor of everybody. This.
Can I say something?
Sure.
And you said very well, but let me add something for you guys that maybe never work with backward dynamics. It's an idea which is very, very nice. It is, imagine that we have a model, like bottom model, which you just have two opinions, black and white. And imagine that half of the population is lattice to make it simple. Half of the population is plus and half of the population is minus. If you run the forward dynamics, a lot of times it's lost because you are looking for people around you who have the same opinion. So here the dynamics of planktons, the area around you typically is very similar to you. So the simulations are extraordinarily slow. The only dynamics happens at the interface. Okay? So the way to simulate that in an efficient way is that you start with some configuration and you go backward in time. So two particles that are similar and nearby, backward in time come from the same one. So that's the way to reproduce the coursing process in a very, very efficient way. You don't lose time looking inside the cluster, not only getting there, right?
Yes, yes.
But you're assuming that the dynamics is temporary.
The dynamics.
That you don't lose anything.
In this case, yeah, in this case, you know what's dynamics is very well defined.
Yeah, but it's not typical. If you are in the book something, going backwards and forward in time, the dynamics is not the same. Because that's purely the same dynamic.
Yeah, well, yeah, because the dynamics here is just code.
Yeah.
So when you copy it together, you can, yeah.
Yeah. In fact, so we have another paper in which we demonstrate that both models are equivalent for at least one for high diffusion. If you have some other particularities, you may lose this equivalence, but we have a huge regime in which it is.
In the lattice is exact.
In the lattice is exact. Is this continuous space, which makes it much more complicated, but it is for high enough. It's just for very, very, very small diffusions in which it's not. But because of strong noise. Strong noise weakness. Okay, so yeah. No, I like that it's more dynamic. So I would like to show a video so that you have an idea of what happens here, because sometimes if you haven't seen before particles in a chaotic advection, could be it's good to know. So here, the gray area is the initial sample, and it evolves backward in time. So you see generations going backwards. And this is the flux. And you see what is particular of chaos, that you have these filaments in time. And they mixes and the mixes in time. But yeah, what we really are interested in these filaments. That is, as we will see, the main cause of this. With time, the communities will be separated eventually, and this is very, very large scale and time. So yeah, the most important part is the initial part. It's what determines everything, almost. So yeah, it's good for you to know how this works. Okay, so back again to this. Okay.
Sorry, what was the gray dot again?
Is where the initial population was, so that you have a reference of what is happening, where they are moving.
How much computational power did you need for flexibility?
A lot. So at OIS we have a computer that is similar to Proteus. So we need that for sure. Because now everything is optimized, and you could have one for each parameter of the flux. The results for the backward with a lot of population, maybe 3-4 days for each parameter, and we will see that we have a lot of parameters to explore all the regimes. So yeah, and that is with the backward model. With the forward, it's crazy. You will need 2 weeks, or if it's optimized. Okay. So then, with these dynamics, we measure what we were interested in, that is the species abundance distribution. So first of all, as I said, we simulate many, many parameters of the flux. And for each, we obtain the species abundance. So quantitatively, we first see that if we put the model with currents and without currents, there are differences, right? In general, these are more steeper. But to quantify this, we measure...
I missed something. How do you measure abundance?
Abundance. So basically, we have our sample. In each individual, we will be assigned to a species. So species 1 will have 2 individuals. Species 2, 5, and so on. So I count. How many species have 5 individuals? So 1, 2, 3, 4. 4 species have 5 individuals. Okay. So you assign 5 individuals, so 5 species have 5 individuals, or 4, I don't know what I said, but 4 species have 5 individuals, and so on. So there are many species which have 1, 2 individuals, and many, like the very big abundances are just for 1, 2 species, for example. So that's a good question to understand what this implies, because if these curves are steeper, that means that our sample is more diverse. So that there are less abundant species, but many species with 1, 2, 3. So many species, much more diverse. Our samples with currents, oceanic currents, than that without currents. This is maybe difficult to see in this figure, but what we do is to represent the exponent alpha of our curves and measure the distribution of these exponents. And what we see is, although there are some areas in which you could not distinguish, there is a clear separation. Because here, exponent alpha, like variance in a difference in this exponent is very, very big, and implies like a lot of differences. So you see that there is a different distribution without currents and with currents. So the exponent is higher with currents. We also measure another diversity pattern, that is the number of species of the ocean compared to the one with ocean, that means with currents.
The distribution is over runs, realizations of the process,
over different parameters. So yeah, one run will be 4 parameters of the flux that makes the jet more longer and different. Yes, also different positions on the space. It's a mess. It's everything. Because it's basically what we have in our sample, right? They took measures in Japan, in many different situations. And yeah, so we also measured another diversity pattern, that is basically the number of species with the sample size. So we see that for currents, the number of species is higher than for lakes, that is lakes without currents, ocean with currents. And it grows in this way with the sample size. So now that we have characterized what happens with currents in our model, we wanted to compare it with the data. So for that, we called our colleagues in OIST, Alex Bousset and Tom Bourguignon, that they are biologists and specialists in sequence data and analyze sequence data, and they help us to analyze our data. So for ocean samples, we used this reference, and for lakes, we used this one. What we observe is this, and for seeing it more clear, For seeing it more clearly, we see this plot. This is our model. The dark one is our model. The light ones are the samples in the ocean and lakes, the real samples. And we observe that both the behavior is the same, and that our predictions are very, very close to what is observed given the circumstances. It's very, very complex biological system, and having this is something very, very good, I think. We also measured, again, the number of species. Oh,
So the data from lakes were out in the expedition paper now?
no, that one. So we wanted to compare that Tara Ocean with the currents, but we needed also data for non samples without currents. So we looked for another source. And, well, this is the data compared to our model, that is the dark one. So the tendency, like the growth with the number of species is also behaves as our model. So once we have proposed a model, we have seen that it predicts quite well what happens in the nature. So now we can think, okay, so now that it reproduces it, let's see why it reproduces it. Now we have the model, we can know what is happening with the sample exactly in time, with the flags, analyze everything. So what we do is to analyze the local finite-size Lyapunov exponent, which is described here, detecting barriers to transport. So this exponent is basically developed to, I don't know if you see the image well, but it's seen that there are some barriers that make the population evolve with these filaments that we saw in the video. And this exponent characterizes these barriers. The procedure to obtain it is a little complicated, so I prefer not to explain it. I had it up a slide, but I think it was a little difficult for this. So just take in mind that we have an exponent for each point at this spatial, in this space. And the exponent characterizes the mobility or the barriers at each point. If the particular, if the individuals are limited spatially by these barriers.
Sorry, the system is homogeneous or it's not?
It's not.
Okay.
We have a chaos. So what we measure, let's see, maybe, okay. So we measure the exponent, measure for each point in this space, with compare with the Lyapunov exponent for that point, particular point. So big Lyapunov exponent says that our sample is more limited in time, in space. And so what we see is that there is a positive correlation. So the higher the exponent or limitation, the higher the exponent of the species abundance distribution. And it has a p-value that is significant. So there is a significant correlation. So what this says is that the exponent is correlated with the advection, the barriers created by advection, by chaos. And it doesn't matter which particular flux you use, if it has chaotic advection. So we have used also a Adriatic model in this reference that is very particular model of the Adriatic Sea. And we observe the same behavior. We also used a vortex model that is very, very simple, the simplest model for chaotic advection, and we obtain the same. To emphasize this, we also measure what happens if the parameter that controls the time dependence of the model is set to 0. So we have the flux, but without time dependence. That means that we don't have chaotic advection. We don't have chaos. And in that case, again, the exponent reduces and goes to the one without currents. So basically, chaos is the cause of this. So what the conclusions can be summed up in these three figures. We first propose a model that is a goal for itself. So basically, we propose a continuous space neutral model backwards in time that is able to reproduce the diversity of huge populations and with in 3 days for very computation. It's very optimal computational. With this model, we predicted the observations in lakes and oceans. And not only that, but we also understood that one of the mechanisms that could be responsible of this increase in the exponent is chaotic advection. So everything has been done, as I said, in collaboration with the Evolutionary Genomics Unit at OIST, Alex Bousset and Tom Bourguignon, and Simone Pigolotti, that you probably know. And well, this has been published here, and if you have any doubts, you can see it. And thank you very much.